Eric?
Is it the bottom little r triple? Because there would have to be an X big R from the father, then that's it.
Yeah, absolutely. It's her right here. So she has to get her two X's, one from mom and one from dad. But if you look at dad's not shaded in, which means he is X big R Y. So does he have an X little r to give to that girl? Absolutely not, and so therefore he can't have that girl, and so that's impossible. So that's what proves that it's not sex linked. And so some places in the pedigree it may work with sex linked, but this shows you that. So that's why you have to kind of do the whole pedigree to kind of make sure that there's no discrepancy within the whole pedigree. So this one absolutely, without a doubt, has to be autosomal recessive. The next one here, this is autosomal dominant. And so what does that mean? It means that everybody who's shaded in has a big R, which means that everybody not shaded in, you know their genotypes for sure, they are little r, little r. So everybody not shaded in is this. And then you can figure out some of their genotypes, like these two have to be big R, little r, because they have little r kids. And her, you can't tell from her parents what her second gene is, but you just know she has a big R. And then these two down here, you know that they have to be big R, little r, because mom had to give him a little r. And then him, he has to be big R, little r, because he had a little r kid. And so autosomal dominant works just fine with this. It cannot be autosomal recessive. Where's the discrepancy here? Where's the proof that it can't be autosomal recessive? Meaning, remember when I say that, the people shaded in are little r, little r. That's what autosomal recessive means. This can't be the case, without a doubt. Shane?
It's two circle kids that are shaded in.
Right here?
Yeah. Because they have to have a dominant trait, but the two parents don't have
The
a
two
dominant.
parents don't. That's right. Absolutely. So it can't be autosomal recessive, and for the same reason, it can't be sex. Whoa, my neck! I strangled myself. My necklace got caught on that thing. So for the same reason, in the same example, you. I'm glad I'm off to the side and not in the video. So she has to be X little r if it was sex linked, and he would be X little r y. So these two girls here would have an X with a big R, and neither one of the parents have an X with a big R, and so therefore that's the same people as examples of why it can't be sex linked. And so then the last one is it does work for sex linked. All right, so, but this last one works for sex linked, but we're gonna see that it also actually works for autosomal recessive. All right, so let's look at this. So if it's sex linked, that means he is X little r y, she's X little r, he is X little r y, and he's X little r y. So with sex linked, you know automatically what the male's genotype is, because the male's genotype is whatever phenotype he has. And so if he has the trait, in this case he's a little r. So she doesn't have the trait, so she's an X with a big R, but we don't know what her second X chromosome is based upon her kids. He has to be an X big R Y, because he's not shaded in. She has to be an X big R, and then her second X has to be a little r, because she had to get an X chromosome with a little r from her dad. That's the only X chromosome he has to get. He over here is an X big R Y. Then we look at their kids. He is an X big R Y. She has to have an X with a big R because she's not shaded in. So she for sure got an X big R from her dad. But her mom could have given her an X with a big R or an X with a little r, so we don't know what her second gene is, and same thing with her. And so therefore it works just fine with sex linked. Let's look at how it works just fine also with autosomal recessive. If it's autosomal recessive, everybody shaded in has two little r's. That means everybody's not shaded in has a big R. So she's going to be big R something. All right, we don't know. He is going to be big R, and he has a kid that's little r, so he's going to be big R, little r. Same thing with her, big R, little r. We look at their kids. These 3 kids, not shaded in, so they have at least a big R, but we don't know what their second gene is as a result of their parents. Looking over at him, he's not shaded in, so he has a big R. He also has a little r. You can either look up at his father, gave him a little r, or look down at his kid, and so that tells you also he has to have a little r. And see, there's no discrepancy with autosomal recessive. So if you had autosomal recessive for that one, it actually is technically correct as well, because there's nothing on here that shows you any different. There's no discrepancy with it. If you had to pick your best guess between it being autosomal recessive and sex-linked, I would choose sex-linked. Anybody know why I would do that? Or have a guess as far as why? If I had to pick one, I'd go with sex-linked over autosomal recessive.
Is it because it says like grandfather, father?
You know what? It's not. It's interesting. Almost every hour, somebody has said that it says the people, but that's not necessarily really the reason. You're right that that's what makes it different. You know, the way that they drew here, but from this we could tell like this person's the grandfather of the, you know, this person. So so that's not the reason. We also have an idea as far as why I would choose sex-linked over autosomal recessive. Well, think about what we talked about yesterday with sex-linked traits. Noah?
Didn't you say it was more common in men than women?
Yeah, it's usually way more common in men than women. And so when looking at this, you see more men than women. So if I had to choose between the two, it would be a sex-linked trait. So this is something you have to be able to do for the test. So you have to be able to take an unknown pedigree and to determine whether or not what's the mode of inheritance. You have to make a hypothesis and be able to test it out just like this. On the test though, what I will do is I will make it so it's clear. It's like this one could be one or the other. I would make it so it only works for one. All right, so that's that. Okay, so it looked like most people did okay on that. Okay, so what we're going to do is we're going to go back to the notes and take just a little bit of notes. We're going to finish 15.3, sorry. Yeah, remember yesterday we skipped 15.2, went on to 15.3. There's just a little bit left in 15.3. We're going to finish that and then go back to 15.2. And one thing I wanted to point out is if you are working on the study guide to go with this, the study guide is from the newer textbook, and the newer textbook, actually the authors decided, like I did, that it makes more sense to do 15.1 and then 15.3 first because it kind of flows better, and then 15.2. So when they redid the textbook, they actually flip-flopped. So the new textbook 15.3 is actually 15.2 in the new textbook. So they flip-flop those two sections. And so when you're doing that, somebody else was confused. They were like, oh, I was trying to do the study guide and it didn't flow the same way, and that's the reason why. So they flip-flopped like the first section, or the second section, the third section, so it's in the same order that we're doing it. Okay? I don't know if that makes any sense. Okay. But if you take a look at it, it might make sense. All right. So. Okay. So. Oops, I'm on the wrong section. I gotta go back. So 15.3 was all about sex-linked genes. And so we left off with, we did some sex-linked problems, and then we did the pedigrees. And so we're on this last thing about sex chromosomes and how sex chromosomes can play a role in your phenotype. And it's this thing called X inactivation. And this happens in female mammals. And so I'm going to use the example of a cat here in just a second, but the cat has the same sex chromosomes as we do. So they have an X and a Y, and the same combination to make males and females. So female cats are XX, male cats are XY. And so what happens is that to get this female, you have an egg cell, the mother's egg cell and the father's sperm cell. Both of them have an X chromosome, because remember when you go through meiosis, males are XY and females are XX. You only give one of your sex chromosomes to your kid. So then sperm fertilizes eggs, and then you get this baby that has two X's and becomes a female. And so this female cat, what happens is as the zygote, the one-celled baby with the two X's, starts to divide by mitosis. Remember, mitosis makes identical cells. So they begin to divide by mitosis, and you get this ball of cells, this embryo, that will eventually develop into a cat. And then in that development process, what happens is at some point, randomly in every single cell in this embryo, one of the two X's for the female becomes inactivated, which means it doesn't work. So a female has two X's, and then in cells randomly one becomes inactivated, so only the genes on the one that isn't inactivated actually work. And so that's what's called X inactivation. So in mammalian females, one of the two X chromosomes in each cell is randomly inactivated during embryonic development. And it's random. So as far as which one will become inactivated. So this plays a role in the phenotype of the cat if the female is heterozygous for a particular gene located on the X chromosome. And what will happen is she'll be a mosaic or mixture for that particular trait. So let me give you an example of what I mean by this mixture of a particular trait. And so the example we're using is calico cats. All right, so this is a female calico cat. She has splotches of orange and black on her. And so how the splotchiness or mosaic, that's what the mosaic means, how that comes about is X inactivation. So what happens, early embryo, this is one of the cells of the early embryo. She has her two X chromosomes. She's heterozygous. So for the trait, for the fur color, she has one black gene on one of her X chromosomes, one orange gene on the other X chromosome. So she's heterozygous. So then she starts to divide by mitosis to make a bigger embryo, and you copy these X chromosomes, so they all have two X chromosomes. Then randomly, in this embryo, some cells inactivate the X chromosome with the black gene on it, and some cells inactivate the chromosome with the orange gene on it. And notice here that it's kind of short and round like this. It doesn't look like the other X chromosome anymore. This is the inactive X. Kind of looks like underneath the microscope, you can't really see like the genes and so on. So what happens is that this is your normal X chromosome. It kind of clumps together, and it looks like it's just a little black dot in the microscope. They call that a bar body. So I'm going to have you write that down. I want you to know that terminology. This is basically an inactivated X chromosome. So what happens is that I'm going to draw up here. So this is your cell. My black marker, I need to get a new one here, so I'm going to use brown instead of black. You have one X chromosome that has the black gene on it, and one X chromosome that has the orange gene on it. So this is your heterozygous zygote. And so then what happens is that they're going to divide by mitosis. So this divides into two cells, and mitosis produces cells exactly like the first, and so now you have two cells. The baby's getting bigger, and so now you have two cells with your two X chromosomes, and then it divides again, and so then you get four cells, and each of these four cells will have your two X chromosomes. But then at some point, I'm not going to keep drawing the cells, so at some point during development, so now this baby is 4 cells big, and then these guys will divide into 8 and so on and so forth. What happens is that one of the X chromosomes in the embryo at that particular time inactivates. So it's just random. So right here, this one could inactivate, the black one could inactivate, the black one could inactivate here as well. Then this one, the orange one inactivates, and let's say the black one over here inactivates again. It's totally random which one does in what cell. And so then what happens is once that inactivation occurs in the cell, then as this baby cat begins to keep on dividing to grow up into an adult cat, when this cell divides now, when this cell divides to make your cat grow, this cell produces two cells exactly like what it was. So these cells here have only the X chromosome with the orange gene on it. These guys only have the X chromosome with the orange gene. This divides, and these guys only have the X chromosome with the black gene, and this divides, and these guys have the chromosome with the orange gene. So what happens is you imagine that every single cell that's a descendant from this cell in the cat will only have the X chromosome with the orange gene. So as these cells divide, and you grow into a big cat, all right, you have splotches or parts of the body that have an X chromosome that produces orange fur, other parts of the body that has an X chromosome that produces black fur, and so on. And so you get this splattering of color, which is what we see here, all right? And it's because of that randomness. And so one calico cat that's heterozygous, because the inactivation is random, like this one might have more orange fur than black fur, but then another female cat that is also heterozygous might have more black fur than orange fur. It depends on which ones are inactivated and where they're at and so on. All right, question?
All right, I got two. Can they ever become reactivated?
No.
No, okay. And then, so if they got rid of the other X chromosome, wouldn't that give them, oh wait, no, never mind. I was gonna say like 45, well, if you're like talking about humans and their chromosomes, they have one less chromosome.
Yeah, technically yes,
Okay.
absolutely.
But it's still considered a pair.
Yes. All right, so I have a quick little animation that's like 50 seconds long that shows this with the cat that I'm going to show you here.
And these cells start off in a female with two X chromosomes. The cell divides, and the embryo is getting slightly bigger, and still, each cell has both X chromosomes active. But in early embryogenesis, each cell will inactivate one of its X's, and one cell will remain with the paternal X as active, while the other one, the maternal one. And now, this process will happen at random, and you'll have almost half of the cells with the maternal X active, half with the paternal X. The embryo will continue to grow, cells divide, all the descendants of that cell, all the descendants will keep the same X active. And you've seen this in calico cats, right? The coat color gene is on the X chromosome, and that's precisely why these cats look like this in mosaic appearance, because of the process of X chromosome inactivation.
All right, so that's just a little animation to help you visualize that. All right, so that's the last thing I want to talk about with sex chromosomes and their role in genetics. So now we're going to go back to 15.2. 15.2 is not about sex chromosomes, which is why I skipped it for right now. So 15.2 is about what we call linked genes. And so this is just another concept that Morgan, the fruit fly guy, came up with. So what we've talked about so far with Morgan is he discovered that genes are located on particular chromosomes. Like for instance, he figured out the eye color gene is on the X chromosome. And so the second thing is that he figured out that genes can be linked together. So every chromosome has lots of different genes. They can have hundreds or even thousands of genes on it, depending upon the length of the DNA and the chromosome. And then genes located on the same chromosome that tend to be inherited together are called linked genes. And so if they're not on the same chromosome, they're unlinked genes. And so what I'm going to have you do, before we do anything more in our notes, is I'm going to have you look at the loose leaf piece of paper that you picked up. the side that says Unlinked Genes. So look at that. So we're going to look at linked versus unlinked. So unlinked genes means the genes are on different chromosome pairs. So we're going to look at some things that, going back to fruit flies, that Morgan did with fruit flies. So if you look here, there's two different traits. There's the B trait, so B plus is gray body for the flies, and B is just black body. What does the plus sign mean again? It's the wild type, right? Wild type means what? What's normal or what's found most commonly. It has nothing to do with dominant or recessive. So therefore, based upon this, do you know what's dominant or recessive, black or gray bodies? No. So I'm going to tell you. Gray body happens to be dominant. All right, so I'm going to tell you that. And then the second trait that we're going to look at, the second trait is the normal versus vestigial wings. So VG plus is normal, vestigial is recessive. So normal is dominant. It also is wild type, but remember wild type doesn't always necessarily mean dominant. Let me tell you what a vestigial wing is. Vestigial wings are short, kind of stubby little wings, and the flies can't fly with them. All right, so they're stuck crawling on the ground. All right, so that's the vestigial wing. So those are the two traits we're looking at. So what we're going to do is you're going to cross two parents. So I give you the genotypes of each parent. So the first cross, the first parent is B plus, VG plus, VG plus. So what does that parent look like? Gray and normal wings, right? And so these genes are located on different chromosome pairs. So what does that mean? So flies have 4 chromosome pairs. So there's chromosome pair number 1, chromosome pair number 2, chromosome pair number 3, and then their 4th pair is they're either XX for female or XY for male. All right, so what we're going to look at is these two traits, and let's say that they're on chromosome pair number 1 and 2, and so they're on different chromosome pairs. So let's make the body color on chromosome number 1 and the wings length on chromosome pair number 2. So for what this means is for the first parent, that means somewhere on this DNA, let's say right here is the gene for body color, and this parent is B plus for that. And then on chromosome pair number 2, let's say right here is the gene to determine wing length, and that parent is VG plus and VG plus. All right, so then that's the first parent. So then the second parent also has chromosome pair number 1 and chromosome pair number 2, but they have the BB and VG genes. And so what I'd like you to do is to cross these two parents and figure out what kind of kids they could have. So then I want you to write what kind of kids they could have here, where it says kids of the first cross. Then what I want you to do with those kids, we're not going to do F2 generation. What you're going to do, it says, do a test cross with the offspring of the first cross and figure out the phenotypic ratio you would expect. So test cross, we learned that in chapter 14. Anybody remember what a test cross is? That's right. So you're crossing the individual with an all recessive individual. All right, that's a test cross. So we use a test cross like for figuring out if somebody who has the dominant trait or something that has a dominant trait is bigger, littler. That's a test cross. So you're going to do a test cross with one of the kids from that first cross and get a phenotypic ratio. So I'm going to give you a few minutes to do that right now.
They can only match each other. So Yeah, but usually it's first, right? Yeah. Wait, so you cross one of these with
So that's just these guys here. So you should have that they all. So, yeah, that's right. Okay, so should I just draw like a share? Okay, that's fine.
All right, so let's look at what we should have here. What?
I feel like I confused myself. I see what it works.
Okay, so the first cross, so this is like how I drew on the board. The first parent has B plus, VG plus, VG plus, and this parent is BB, VG, VG. So remember when this parent right here goes through meiosis, they're going to pass on one chromosome number 1 and one chromosome number 2 to their kids. So they could pass on this B plus with this VG plus, or this B plus with this VG plus, or this one with this one, or this one with this one. No matter what though, they're going to give a chromosome number 1 with a B plus and a chromosome number 2 with a VG plus. So therefore there's really only one combination that this parent could pass. So I made a really kind of a 1 by 1 Punnett square, because this parent here I just put could pass on this combination, B plus and VG plus. This parent, you could have wrote it 4 different times, all right, made the big Punnett square with 4, all right, and then this parent here, there's only one combination as well. They could pass on this chromosome number 1 with this chromosome number 2, B and VG, or B and VG, B and VG, or B and VG, but they're all B and VG, so therefore the only combination for those 2 genes is this combination. So therefore, the only kind of kids that these 2 parents could have are heterozygous for each trait, B plus and B, VG plus and VG. And so all the kids are going to show just the gray bodies and the normal wings. So then the next cross, so that's the kids there. So then the next cross, you're going to take that kid and do a test cross, which is crossing them with all recessives. So BB, VG, VG. All right, and so that's what we're going to do here. So this parent here, when you do your test cross, can pass on 4 different combinations to their kids. The B plus and the VG plus, so that's one combination. The B plus and the VG, which is another combination. B and VG plus, and B and VG. So those are your 4 combinations here. This parent, for the same reason as the parents that we talked about up here, can only have one combination. They're gonna pass on a chromosome number 1 with a B and a chromosome number 2 with a VG. So I only wrote it once. You could write it 4 times, and then you just have the same thing answer there. So therefore, when you fill this out, your phenotype ratio is a 1 to 1 to 1 to 1 ratio. Okay, that's what we got. All right, so now what I'd like you to do is, if you flip your sheet over, we're going to look at these same traits, but now we're going to treat them, instead of as unlinked genes on 2 separate chromosomes, what we're going to do is make them linked genes. Linked genes are on the same chromosome. So if we look up here, that means that, let's say the wing length is not on chromosome pair number 2. We're going to treat it now that wing length is on chromosome pair number 1 with the body color as well. So both of those genes are on the same chromosome. So let's say right here on the DNA is the gene, the VG plus gene, and this is VG plus as well. So notice that you're doing the exact same cross. You're starting out with parents that have the exact same genotype, but now we're going to treat these as linked on the same chromosome. And I think with the linked genes, it's helpful to draw it out, so you can see it on the same chromosome. So these guys are on the same chromosome because it matters eventually in what combinations they can pass in their kids, in their egg and their sperm cells. So this is one parent on chromosome pair number 1, and then the second parent is also the same as before, little b, VG, VG, except they're on the same chromosome. So the second parent also has a chromosome pair number 1, and the second parent, though, in the same spot as the body color, instead of B plus, they have B genes, and then where the piece of where the wing gene is at, they have VG and VG instead of VG plus. So this is one parent, this is another parent. We're going to cross those and see what kinds of kids that we can have. And then from that, you're going to do a test cross to see what kinds of kids could result as a result of that, doing a phenotypic ratio. So go ahead and do that right now. So you're treating them as linked. No, you won't be able to tell just from the text. So with this, it doesn't matter.
So that was the process. So that process is just B and G. Like how.
Oh, yes, yes. I think I don't know.
That's it. Yeah, like that's like how you set it up.
So how do you set it up? This might So this parent is only has one. However, it has this one. But this parent. Just do this right now, alright, and then we'll work from there. So yes, so with them being on the same chromosome, there are pairs together, so that limits like choices, but in this case here, both the chromosomes are these two genes, so therefore, either way, it's the same thing, right? And the same thing goes with this one, so you can do a one by one.
This one's the same thing, because you can only give this one.
Right, so then there would be two
Until you
combinations,
these
so
two
there would be the B plus and
test
the G
cross,
plus and the G.
Okay.
so
So,
they could either
is
give
this gonna
this
be-
one,
So it makes smaller chromosomes because there's less.
and then that's what you only do.
So then this is, so you can,
And I can't really,
and then you can do a test cross, yes, yes. So they have.
yeah,
So one.
well, that's it. So yeah, the
Now you
first,
take this one, so draw out the chromosomes.
the bottom one is the top position. The first cross, they're all gonna be this. But then, once you cross them, you're gonna have like, either have this chromosome or this chromosome. And then with this cross, you're gonna have this chromosome, this chromosome.
Okay, I'm like,
So it's like.
wait, so this is G plus and then B.
You're doing it.
And you know, these have to be on here together because this parent was on the same chromosome. See this one? Yeah. Does that make sense?
I was
Right, so we're doing the order, but sometimes we don't pay attention to how they were on the parent.
Yeah, that's what.
Yeah, so then. So now we're.
So now you have to cross these. That's the sweet. Right, now you have to cross it.
Oh.
All the stuff. So we're not have yours crossed, like, so these two.
Yeah, thank you.
But then you have to, for the other parents, you have to get the all recessives. Okay, so this parent is going to pass on this chromosome or this chromosome. So therefore, but the combinations are the same on both of them. So B plus, G plus, or B plus, VG plus. So therefore, this parent can only pass on the B plus and VG plus to their kids. That's the only combination. For the same reason, the only combination that this one's going to pass on to its kids is a B and a VG, because they're stuck on the same chromosome. And so remember, they only get one of each chromosome to their kids. So therefore, this could be like just a 1 by 1 Punnett square where this parent would pass on. The only combination is B plus and VG plus. This parent. Does that make sense?
Yeah.
Okay. Okay, so you're right here. You're right here. This is right. It's just this cross is wrong. So this is you're crossing two.
Oh yeah, and then I told her afterwards because I couldn't go there. And Mount Zion.
I didn't know so.
So yeah, and one thing I can give you.